This is an animal called Trichoplax. It's the smallest, simplest free-living animal. It's actually smaller than some unicellular organisms. This guy, which we found in the Otago Harbour, is only about 100 microns across. That's a lot smaller than some marine amoebas. But it's a multicellular organism. It consists of 2 layers of epithelium containing thousands of cells with a squishy layer of other cells in between. So it's a collapsed epithelial ball resembling a collapsed blastula. Now, despite it being so simple and not having any sense organs or muscles or nervous system, it has quite interesting complex behavior. It also may be closely related to the ancestor of all animals with nervous systems, and so by studying this animal, we can find out something about how nervous systems evolved and how nervous systems work. We'll start by looking at a simulation of Trichoplax feeding behavior. So this model is going to move across and eat those bacteria. Everything in the model is based on Trichoplax, but the model's actually much simpler than Trichoplax. So think of it as a Placozoan. It's a model of some simple ancestor of Trichoplax. When we've looked at the feeding behavior, we'll go back and look at how it works and what that might tell us about how nervous systems evolved. So the animal's crawling along on its cilia. It encounters some bacteria. It moves over the bacteria, releases its digestive juices, digests the bacteria, and moves on. So let's break that down. So this animal has ciliated cells on its ventral epithelium, and it moves by beating those cilia. So it's literally walking along the seafloor on its cilia, which are all beating, moving it to the right in this case. Real Trichoplax in a uniform environment will just move around at random until they encounter some kind of food. I've given this guy a shove towards the bacteria. When the animal encounters some bacteria, some chemosensory cells near the edge of the animal, literally taste cells, taste the bacteria. They release a neurotransmitter which causes nearby ventral epithelial cells to beat their cilia in the direction towards the edge of the animal, and this causes the animal to move in the direction of the clump of bacteria. As the animal moves over the bacteria, those taste cells map out the locations of the bacteria underneath the animal. At some point, as the animal crawls over the bacteria, lipophil cells release their contents, which is acid and digestive enzymes, into the space underneath the animal. The bacteria are digested, they're broken down into molecules. Those molecules are taken up by ventral epithelial cells and by fiber cells in the middle layer. And when all the bacteria are digested and the products are taken up, then the animal moves on. Now, if we go back to this step, there's something quite interesting happening here. Right, those taste cells are mapping out the locations of the bacteria. So the bacteria form a pattern in the world, and that pattern is being mapped onto a representation on the ventral epithelium. So this animal is forming a representation of a relevant part of the external world in an epithelium of its own body. Second point to notice is that the lipophil cells are not activated at this stage. Right? So it's mapping out the locations of the bacteria, but it's not eating them. And in the model, what's going on is that if you look carefully, there's a central zone here that contains the lipophil cells, the digestive cells, and then there's a zone around the outside that doesn't contain digestive cells, it only contains sensory cells. So this region out here is essentially giving a heads up to this region in here about what's coming up, so what's out there. And in the model, there's just an inhibitory signal here. So this cells in this layer here are responding to the presence of the bacteria. They taste the bacteria, but they're sending an inhibitory signal into this central region here so that the taste cells here are not activating the lipophil cells. So as long as these cells in this outer zone here can see more bacteria coming, a sufficient number of bacteria coming up, they'll inhibit the Trichoplax so that it doesn't release its digestive juices. And there's a good reason for that, and that's because these digestive juices are expensive. You don't want to be releasing these enzymes and acid into the environment if there's not a sufficient number of bacteria there to justify that expense. So when Trichoplax is over a sufficiently large number of bacteria, it will stop, release its digestive juices, digest the bacteria, and then move on. So this mechanism is a simple mechanism for making a decision about whether or not the animal should pay that cost in order to feed, or it should just carry on moving. And this is all done without neurons. This is all done by chemical transmission, but the chemicals that are being used, that are diffusing from one cell to another, are neurotransmitters. They are the same neurotransmitters that are being used in our brains. So just to recap, this simple organism is mapping the distribution of food in its environment. It uses that map to guide the release of digestive enzymes to efficiently digest that food. And it has a specialized region of the map around the edge, which draws the animal towards the food and tells the animal what's coming up so that it can make a good decision about when to stop and eat. This is remarkably similar to what we do, right? We have sense organs, and our sense organs tell us what's out in the world. They give our stomach a heads up about what's coming, and they guide our movements towards the food, and they help us to make decisions about whether we should stop and eat the food or whether we should carry on and look for something else. So this outer region, the outer zone of the animal, is acting kind of like a nose or a tongue, or like eyes and ears, or like the antennas of insects, detecting things in the environment, guiding the organism towards things that it might want to eat, and helping the organism to make decisions about whether it should actually eat them. It's all done without neurons, but it's done using the same chemicals, just using chemical diffusion, using the same chemicals that are in our brain when we make these kind of decisions. Trichoplax turns out to have several different kinds of sensory cells, so neurosecretory cells, cells that detect some chemical, something in the environment, and release neurotransmitter. In the model, the neurosecretory type 2 gland cells tasting the bacteria and guiding this behavior. There are other types of gland cell, neurosecretory cell, concentrated in this outer region, the marginal zone of the animal, which also contains crystal cells, so cells that contain aragonite crystals, which happens to be the same material that's used in the statocysts of invertebrates and the eyes of invertebrates. The compound eyes of invertebrates use this material as their lens material. Now, interestingly, although Trichoplax doesn't have arms and legs and antennas and eyes and ears and noses and things, it just has this tissue, it does have the developmental genes that in bilaterian animals lay out the organization of those structures, the homeotic genes. So there's a HOX gene, the TROX2 gene, which is a precursor of the gene that lays out the anterior-posterior structure of our nervous system. There's the BMP and cordon system, which in animals like us lays out where the central nervous system is going to develop and where the digestive system is going to develop. It has this PAX-B gene. The PAX-B gene is a precursor of the genes that lay out where the eyes and ears of vertebrates are going to appear in the developing embryo. And that PAX-B gene seems to label the area where those crystal cells turn up. There are other transcription factors expressed in Trichoplax. The spatial pattern of expression hasn't been worked out well enough to draw into this map, but those genes include the antennapedia genes, so the genes that in vertebrates are responsible for giving the identity, segment identity, and the identity of antennae and legs and feelers and things like that. So although this outer region of Trichoplax doesn't have any morphology resembling the sense organs and the sensory appendages of other animals, it behaves the same way. It performs the same function, and it has the same developmental genes expressed in that region. So to put it all together, this simple organism is capable of sensing and perception. So it takes the sense data and it maps that into an epithelium, into an internal representation in an epithelium, and it uses that representation to make decisions and to guide its actions in relation to what's in the real world. So it's kind of a microcosm of everything that we do using our sense organs and our brain and our muscles, except that it's not using any neurons. It's doing it all by chemical diffusion signaling. And it seems to be using, at the biochemical level, at the molecular level, it seems to be using the same mechanisms that we're using, apart from not using neurons. So I've gone on long enough, so we'll stop here, but we'll finish with a couple of questions. So the first question is, so this animal is capable of perception, but it's only perceiving the world that's directly in contact with its body surface. It can't perceive anything beyond the edge of its body like we can, so we can perceive things off at a distance, so we can map the world on a large scale compared to the size of our body. So question number one is, can an organism like this do it? Can this model be extended so that the organism can see things in the outside world beyond the edge of its body? And the answer to that is yes, and it's actually really easy to do. And I'll make another video showing how that can be done. And the second question is, well, if you can do all of this without using neurons, and you can, as I'll show in the next video, why do we have neurons? Why do we have brains? And what are all those neurons doing in our brains? And the simple answer to that question is that it's a scaling issue, that chemical diffusion, that these reaction diffusion processes that are implementing this sensing and perception and decision making in this animal only work on up to about a few hundred micron spatial scale. Once you get larger than about a millimeter, it just becomes glacially slow. It just can't keep up, so it's just not fast enough. So the simple answer is that neurons are the solution to a scaling problem, that they enable chemical brains to operate on a large scale. But that's all I've got time for today. I will be making some follow-up videos to follow up on those questions and talk some more about brain evolution and brain function, and to fill in a lot of the details that I've skipped over in this video.